Okay. I do have research support from NIH and a small research grant from Bayer Pharmaceuticals, and I've been a consultant. Oh, not a major stockholder. That is the wrong line. I've been a consultant for Bayer and Biogen IDEC. I'll have to change that in the official record. Sorry. I was very late last night when I did this. And as has been so eloquently explained by Professor Adami and Bernard, sickle cell is basically sickled red cells that can stick and clog up the blood vessels. And the primary clinical manifestation is hemolytic anemia, life-threatening infections. And what Francois, I think, has really... so I cannot get a pointer without the thing going forward. Sorry. So Francois really pointed out that I had thought of this more simply, that vaso-occlusive disease is more the sludging, inflammation, coagulation, and that the arterial disease led to the CNS stroke. It's quite complicated, and I'm going to see if I can get the pointer to work. Okay, there we go. And I think that the vascular endothelium becomes quite injured and abnormal in sickle cell disease, and thus the vascular emphasis in this talk. There's many immune mediators, mast cells, iNKT T cells, neutrophils, which I think are very primary drivers behind some of the inflammation. Platelets are key and likely involved in the end stage and are quite activated by platelet experts at this session and at this meeting. There's also, with the hemolysis, in the afternoon, Greg Cato's, or in the second session, Greg Cato's going to talk about results of hemolysis, hemoglobin release, reactive oxygen species, and decreased nitric oxide bioavailability. And these can lead to chronic inflammatory pro-oxidant damage to the endothelium. And then intermittently, and at times where we have full vaso-occlusion, and there's potential roles of the coagulation system through tissue factor and ultimately fibrin deposition. We like to hypothesize that acute pain is with the acute vaso-occlusive event. Chronic pain is a whole different beast, and I think we're just beginning to understand that, and not the topic of today's talk. Greg's speaking later this morning, and when I showed that figure probably about a decade ago, he came up to me and said, I think I have a better way of thinking about this. And this is in the upper red part, the viscosity vaso-occlusion. And that's how I always thought of the microvascular silent strokes. But Francois has pointed out very elegantly, we also still have to think of the arterial phenotype. In the lower blue corner is your high hemolytic rate, release of red cell contents, and more on the arterial injury. My role today is simply to point out the mouse models of sickle cell disease, and I think it's real important to always try to recognize when it mimics man and when it doesn't mimic man, which could lead you down the wrong path, which has been a critique of any mouse model. For the first decade or so, we had several varieties of mouse models, SAD, NY1DD, and all these had a mixture of human and mouse globins. Mouse globins are very anti-sickling, so they tended to have less hemolysis and more variable pathology. However, they were key to some seminal findings and understanding of sickle cell disease, so did play and still play a critical role. In 2014, the two most commonly used severe models, one is the Burke sickle model from 1997, which is 100% human sickle hemoglobin, and it's a knockout with the knock-in of the human globin genes on a different chromosome. And it has a high hemolytic rate and severe sickle cell pathology. So severe it's hard to generate lots of mice. And in 2007, Tim Towns described a new model that he also had one in the initial one that was similar to the Burke mouse, but he has a new model where he knocked the human globin genes into the proper promoter locus control region in the mouse globin genes, and they also have 100% human sickle hemoglobin, and I think we're still learning about what exactly the phenotype is, but it clearly is severe sickle cell disease. And if you compare them head to head, yeah, I don't know if I'm going to have a pointer. So if you compare them head to head, the hemoglobin levels are pretty similar. While the Townes mouse tend to have a little bit higher hemoglobin levels, their retic rate is even much higher. And I'm not quite sure what drives that in this mouse. The Burke model has had the problem or potential usefulness that it's also the beta globin is less than the alpha globin for the human gene synthesis, so they're low beta thalassemia, so their MCV is lower. The high one in the Townes might be in part related to the high retic count, which increases the mean cell volume, the red cell mean cell volume. Both have nice evidence of vascular congestion, which I think is the equivalent of vasoocclusion. That's still up to debate. They both show hypostenuria, where sickling in the renal medulla in very early childhood makes it very difficult to concentrate urine, and so the young children have to go to the bathroom and drink more because they cannot concentrate their urine. The mice express this very well in both those models. Both show tissue ischemia or infarcts, and splenomegaly, which is a little different than we find in man, in part because in hemolytic anemia, mouse models have their extramedullary hematopoiesis primarily in the spleen. Strain background, the Berkeley model is on a mixed model, so we try to do sibling controls with the Berkeley hemoglobin A. And the Townes model are 50-50 black 6-1-29, so you can actually have a more comparative control. To grow either of these mice on a homogeneous well-bred cross, such as just black 6, it makes it nearly impossible. They need some of the genetic variety for many of the labs, including my lab. New York has been able to do this. What do the mice look like? So to the far right is mama mouse, and then there's 4 pups, and this is at about 28 days when they're supposed to be weaned, and you can see that one of the 4 siblings is much smaller. That's a sickle mouse, so it sort of mimics what we see in humans, where the patients tend to fall around the 5th percentile, but grow, and eventually they catch up and become similar in size. So human sickle cell disease is clearly an inflammatory disorder with elevated white counts, activated neutrophils, increased invariant NK T cells. There's also measures of endothelial injury with circulating soluble VCAM, E-selectin, P-selectin, and C-reactive proteins, cytokines, leukotrienes. Most inflammatory markers are up at baseline. Many tend to go up further during an acute painful episode. Sickle cell is also a prothrombotic disorder, and this is the subject of Kenny Taga's talk in the second part of this talk, and I'll let him go into this further, but I want to show how my mice data line up with Ken's data. And so there's increased thrombin generation. I'm going to pretty much focus on that, decreased protein C levels. And as this whole meeting can tell you, that there's a lot of crosstalk between the inflammatory and the coagulant pathways, where thrombin signals via PARs, increased cell adhesion, PAF expression. There's increased tissue factor activity, which I see Nigel Key's in the room, and he's done some of the seminal contributions. And also there's increased consumption of protein C that's been described for many decades, that the protein C levels are just a tad bit lower in sickle cell compared to humans, and I don't think it's a genetic deficiency. My hypothesis is increased consumption. And so protein C, APC has many anti-inflammatory effects and endothelial health effects, and so I think to have a limited resource for generating APC is a problem that further contributes to the endothelial dysfunction in sickle cell disease. And to put this together, it has to come back to the red cell, because if you don't have hemoglobin S, you don't have any of these findings. And the sickle red cell clearly interacts with the endothelium, and it kind of sets up a vicious cycle of thrombosis and inflammation, as well as when it lyses and the pro-oxidant products. And I think there's a lot of novel therapies that could intersect at any of these things that many labs are doing, and at the end I'll come up back up against a couple of them that I think the mouse models have helped us with. So the first question is, do mice with sickle cell disease have increased inflammation and coagulation activity? If they don't, they're not a good model to study those pathways because they don't model human disease. In the sickle mice, here's a section of liver, sickles, upper left, control is a healthy liver. We perfuse the mice, so they shouldn't have any red cells trapped. And what's circled is a nice infarct, not nice for the mouse or the human, and it mimics what we can see if we do autopsy studies on humans with sickle cell disease. And the kidneys show this, all of the organs pretty much show this. And it's unique to the sickle mouse compared to the control. If you grind up the organs and look at organ-specific fibrin deposition, in every organ, including the brain, there's not many mice that have increased fibrin in their organ. We see that the sickle mice have it compared to that. So there's ongoing thrombosis and fibrin deposition. We use an antibody that doesn't detect full-length fibrinogen, so it's clearly been cleaved and potentially cross-linked. Thrombin, antithrombin, which is hard to measure in mice. You have to have a very, very quick, clean draw. But compared to our control hemoglobin A mice, the sickle mice have increased levels of thrombin, antithrombin. So they show increased thrombin generation. And protein C, similar to human levels, almost nearly identical. There's just a modest decrease at baseline compared to control mice. So I'm going to switch gears a little bit to inflammation. So in humans, sickle cell alone is pro-inflammatory. What if you have a second pro-inflammatory insult? And in inner cities such as St. Louis and Milwaukee, we've shown that approximately 20 to 30% of African Americans have asthma, children. And so that suggests, given the ethnicity of African Americans, that 20 to 30% of our patients also have asthma. And Mike DeBon's looked into this further. And one of his first studies was looking at, in the CSCC database, is you check the box off yes or no for asthma. What's the impact on mortality? And he found that if you have comorbid asthma, just doing a retrospective chart review, that there's an increased risk of death. So it's such that you start with one inflammatory, you add a second one that shouldn't affect lifespan, and it does. So we went into our mouse model to see if we can mimic this. This is with Mike DeBon. And we chose the subcutaneous ovalbumin. We waited 2 weeks, and then we aerosolized ovalbumin after they were sensitized every other day for 10 days, and then we had a traditional mouse asthma model. However, we ran into trouble. So in the first 2 weeks, all of our mice, except for the sickle sensitized, which is the green upside down triangle, many of them died. And we made sure that we didn't start counting deaths until about 24 hours after. That's why there's no carves there. So it wasn't anesthesia or procedure deaths. So you just add the ovalbumin implant, which should be fairly benign. We have increased death. The mock surgery did not do this. So then we go to aerosolization. You can see that same upside down triangle. I renormalized it back up to 100%, and we looked at that, and we had major mortality to the point where we were having trouble getting enough mice at the end. So we did this low sensitization, and we were able to come up with enough mice to study at the end. The high sensitization is actually the normal way to make asthmatic mice. The low sensitization, we just cut the aerosolization time in half, because that's when they started looking like they were having trouble breathing. And if you look at the lungs pathologically, the high sensitization on the wild type on the top row was a nice model of asthma with increased eosinophils. We had the basement membrane thickened, and we quantified all that, and we found we had a good model of asthma. Similar for the hemoglobin A mice. We wanted to try it on our mixed background to see if there wasn't a background effect. For the sickle mice, the low sensitization, when we added everything up, was the equivalent of the high sensitization in wild type and hemoglobin A, suggesting that you add sickle cell in, you're pro-inflammatory with a much less of a secondary trigger. And this kind of does this. I don't think we never could do this in a human, but we gave a low dose of LPS, endotoxin, and at a level that if 50 micrograms per kilogram was a 50% lethal, we tried 8. And Lewis Su had previously published this with 10. And none of our control mice, trait mice, hemizygous mice, AA mice, wild-type mice, none of them died. And with the sickle mice, when we first tried 10, they all died within 4 hours. And by backing off to 8 micrograms per kilo or per gram, we had nearly all of them die at 48 hours. Suggesting the mice really are sort of very much on the edge with inflammation. I believe it's similar to humans, given the asthma data, and that you don't need very much to tip the tide over and cause much more morbidity and mortality. And I think we have to keep this in mind when we're designing studies. And I also think that shows that the mouse model is a good model for human disease. Okay, the big question is, does acute vaso-occlusion affect coagulation activity and inflammation in sickle mice? So there's a lot of debate, what's the best model of vaso-occlusion, because the mice can't tell you when they're having problems. They're hard to monitor by repeated blood draws and this and that. So our plan is simply to induce sickling, because we know that hypoxia is the only time that sickle hemoglobin will sickle. So we found conditions where the mice will 100% survive when we place them in a hypoxia chamber, 2 hours for the behavioral studies, 3 hours for most of our other studies at 10% FiO2 to induce acute sickling. And the reoxygenation is 1 to 4 hours to permit time for tissue response. We've also looked the next day. And Bob Hebel's group actually initiated a lot of these studies, and we're modeling after his studies. And this induces sort of a ischemia reperfusion phenotype in the sickle mice. It really mimics a lot of the changes that we see in man, and oxidative injury, inflammation, and you can read many of Dr. Hebel's articles on this. In our hands, in our model, the control mice, whether they be hemoglobin A or wild type, have no measurable changes following hypoxia-reoxygenation. Similar to humans, though, humans with trait go to high altitude, they can have acute sickling. The mice with trait, we can induce sickling in this environment. What do we see if we perfuse the mice? I would really like to have a pointer. Okay, so we perfuse the mice. So this vein is well cleared out, and a lot of these microvessels are clear. And this is normoxia, so this is at baseline. This central vein has loosely packed red cells, and around it looks more red because a lot of the microvessels are packed. So to me, that's my definition of vascular congestion. You've perfused the liver. Normal mice have no blood in their liver, and despite that, there's retention of blood. So I think this is vaso-occlusion that's coming and going intermittent. After 3 hours of hypoxia and from 2 to 4 hours of reoxygenation, when we rinse all the vessels, we see a very different phenotype. One is we get very packed veins, and then surrounding them, this lighter area is an ischemic hepatocyte. The nucleus looks pretty good, and many of these recover because by the next day I don't find increased infarcts. But there's clearly ischemia around central pack. And so I think that this is a good model of acute sickling, hypoxia-induced vaso-occlusion, and we use this as our model. In the lungs, we found similar findings where normoxia and hypoxia in the control mice, we can pretty much rinse the lungs free of all blood. In the sickle mice, we have patches where all the vessels are clear, and where there's loosely packed red cells. After hypoxia, it gets greatly advanced in the lungs. As far as, is this a good model for inducing acute increases in inflammation? If you look at normal AA mice versus baseline normoxia sickle mice, there's more liver ischemia because we don't see that in normal mice. The ALT at baseline is up. That's a little different from humans. So I think the liver is at higher risk of sickling complications in the mice compared to man. So that's one difference. Thrombin, antithrombin at baseline is up in our mice with sickle cell disease versus the control AA mice. And so that mimics our others and MAR findings in humans. The white count is nicely elevated in the teens compared to a normal mouse white count, and that's very similar to what we see in humans with severe sickle cell disease. CRP, which in mice is serum amyloid protein, is also quite elevated in the sickle mouse. At baseline, it's up in humans. Some of the cytokines, gamma interferon, IL-6 are 2 that I've shown here at baseline, are modestly up. And then after inducing acute sickling with hypoxia-reoxygenation, we have a further increase in acute ischemia. The ALT can go way high off the top. Thrombin, antithrombin, so there's an acute burst of coagulation that settles back. Everything settles back to normal the next day. So our conditions are not permanent, sickling with permanent organ damage. Same thing with the white count, it tends to settle back down. The CRP goes up. We didn't test that for returning. And the cytokines, these 2 at least, can go up and then tend to return. So it looks like we have a transient, timed, acute sickling model. And so we use this to study that. You can now tie this in your thinking when you see the afternoon talks. So Greg is going to be talking about the arterial dysfunction. Francois has already done a beautiful job pointing out that the arteries are different. This is a whole different pathology in my mind, although my thinking about silent stroke is now completely different after Francois' talk. And this is more about hemolysis, nitric oxide bioavailability, vasodilation, and more oxidative injury. And I think it's more to explain a very different vascular dysfunction in sickle cell on the arterial side, and it's an important one. And also stolen from our speaker from this afternoon is Greg Cato, and that just kind of shows that when the red cells lyse, there are many other things that come out, and I think I'll let Greg spend more time on this. So how about in the mice? Well, in the mice, we're looking at hemolysis in sickle cell disease. Nancy Wandersee, who's sitting in the front here, also is a hemolytic mouse expert, and she has these mice with severe hereditary spherocytosis where the lifespans of 1 day, they're close to 99% retics and are more anemic. So their model of pure hemolysis with no sickling. So how do they compare? I just have a couple more slides and I'll end early. Her mice at birth are very anemic, and this is actually a normal newborn mouse, pink, 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 and then she lined up from several things because she has higher mortality, and the white ones are her mice, so she can do a genotype phenotype on day 1. And if you take, I was very interested in stroke, and if you take the fascialis artery, which is a branch off the carotid, put it on a pipe, dilate it or preconstrict it, and then try to dilate it with acetylcholine, with increasing amounts of acetylcholine, you should normally have nice dilation. So that's what our control mice do, both in Nancy's colony and the sickle mouse colony. However, the sickle mice act a lot like the severe atherosclerosis mice, so the ApoA1 knockout mice, the LDL receptor knockout mice with a Western diet, where they have poor vasodilation. In my original study, it was just a flat line because I think they were 12-month-old mice. But we see in further studies that we see a little dilation at the end, very, very similar to hereditary spherocytosis. Nancy's mice also stroke at a much higher rate than the sickle mice do. So I think that it suggests that Dr. Cato and Mark Gladwin and his colleagues are on the right line. Just an interesting study that we published quite some time ago is that if we, and this is the same fascialis artery, and if you, and this is where it's a flat line because they're old mice. I thought it was a stupid study, so I gave them my oldest mice. We treated with them with this 4F peptide, which is an ApoA1 mimetic peptide, sort of like the backbone of HDL or good cholesterol, and it can pull out oxidative lipids. And I didn't think it would work because cholesterol and HDL cholesterol is low in sickle cell disease. And so we treated the mice with this peptide for 3 weeks, and we almost normalized, well, more than halfway normalized this vasodilation defect, suggesting that if you can improve the oxidative injury of the lipids, then we can have some improvement potentially in sickle cell disease. And I know that other studies have gone on with this, such as statins and other things that have been thought about in sickle cell disease. The other thing that's Mark Gladwin and Greg Credo have done seminal studies is just pointing out that mild elevated pulmonary artery pressures as measured by a screening cardiac echo, where you're not proving it's pulmonary hypertension, but you do put a huge risk factor for death. 30% of young adults have this risk factor, high TR jet velocity, and these are the adults that tend to die in the next few years. Do the mice have it? It's not a good model if the mice don't have it. And what we find is, we use right ventricular systolic pressure as an estimate of the pulmonary pressure, and in normal controls versus sickle mice, we do have a modest elevation in the right systolic pressure, suggesting pulmonary hypertension, actually proving it in the mice because we stick the needle right in. And Nancy's mice also have this, suggesting this really is a hemolytic event. If we measure right ventricular hypertrophy as a secondary marker, we also show evidence for that when we normalize for body weight. As far as endothelial injury, soluble VCAM is a good marker that's up in human disease and E-selectin, and the sickle mice have elevated levels suggesting chronic endothelial injury for both E-selectin and soluble VCAM, so there is chronic endothelial injury. The hereditary spherocytosis mice also have this. But you can see that the sickling and the microvascular ischemia reperfusion probably make this worse. So the sickle mice baselines are higher than the HS mice baselines. So this is my last slide. And mouse versus man. So is it a good model or is it a bad model? Has it led us right or wrong? So from an inflammation, coagulation, and I didn't show my videos or adhesion data, but from an adhesion standpoint, I think it's a good model. It's a model of severe sickle cell disease. And I think that anticoagulation and anti-inflammatory therapies warrant further thought. Kanataga is doing anticoagulation therapies. Others have done anti-inflammatory therapies, which is beyond the scope of this talk. And also there's some clinical trials for anti-adhesive therapies. And so I think the mice have helped us understand this better and provide some important preclinical studies. For the arterial dysfunction, I'm going to leave that more in Greg's basket, whether he thinks the mice have led us down the right path or the wrong path. Because the arginine trial, inhaled nitric oxide, and sildenafil have had mixed outcomes and haven't gone forward, and maybe the mice have shown more than what's in man, or maybe it's just too complicated to have such a complicated disease, have a single intervention that can improve it without moderating the sickle hemoglobin in the first place. And so with that, I think we can close my talk. This is acknowledgement of the people at the Wisconsin Sickle Cell Program, 8 miles up the road. Paul Scott leads the program. We have a lot of interactions between the bench scientists, which are below the bottom line, and the upper line, which is a mix of outcomes, clinical and basic research, who also get called to the emergency room when the patients show up. Okay, so I'm going to take questions for all three at this time, and thank you for your attention. And we'll probably only spend 3 or 4 minutes because we are over.
Yves Gruel from France. Thank you for these presentations. Red blood cells are a large source of ADP, and I'm wondering if there is a place for antiplatelet agents inhibiting PY12 receptors in the prevention of complications due to vasculopathy in SCD.
Yes. So there is an interest in platelets. Clearly the platelets are activated. The antiplatelet therapy drug trials to date have going back as far as aspirin and many of the other agents, haven't shown an effect on pain. But if the platelets are more important in the arterial disease, I wonder whether it should be primary prevention for stroke, potentially in countries that cannot afford transfusions and TCDs. I think they might have looked at the wrong outcome, but none of the platelet trials in humans have shown an improvement in acute vaso-occlusive painful events. But it's still an area of high interest, and I hope people in this meeting will become more interested in this. Barry Kohler's also looked at platelets in sickle mice. Okay, and I think just
Sure,
two
I'll
questions so we can get to our
I'd just like to add to that answer. There is an active phase 1, phase 2 trial going on prasugrel that's been reported at several meetings that is looking promising, actually, for the sickle cell pain phenotype.
Fantastic.
Just to expand upon that, it's actually a phase 3 trial. I'm with Eli Lilly, and there's a phase 3 trial with prasugrel, which is obviously a P2Y12 antagonist ongoing. And just to contradict you a little bit, there is actually some data on antiplatelet agents from back in the 80s. Aspirin isn't really relevant because it doesn't inhibit ADP-induced activities very well.
There you go.
Whereas there are 2 studies with ticlopidine, which is a P2Y12 antagonist. One was a very small study with 9 patients. The other had a few hundred patients, and there were signals for reduction in
Right.
in
So there were
painful crisis.
biomarker improvement, but it didn't, and it was too small to tell clinical outcomes. So,
So
the
there's-
biomarkers did improve with those 2 studies.
But there was a pain study by Cabanas in the 80s, and that
Okay.
showed some encouraging signals.
Okay, this is why we're doing this. Okay, and I think that since we're late, we'll adjourn and come back and start promptly at 10:15. Thank you very much.